Okay. Hello students, welcome to Aakash. I'm your botany faculty Aman Vishwakarma. And today we are here to discuss the AIATS plus phase 2 paper of mock test 4 that was conducted on 9th of Feb 2025. And from the previous session you already know we are here to discuss those questions where most of the students have made mistakes in. And for this session I'm going to discuss the questions from these topics. Sexual reproduction in flowering plants, organism and population, plant kingdom, morphology in flowering plants, cell the unit of life, and photosynthesis. So let's start our first question. Question number 95. Thorns found in citrus and bougainvillea are. So this question belongs to morphology, and the questions asking us about whether the thorns are formed or are vegetative structures or they are where they are originated from. Okay. So let's see the concept. As you already know that there are various modifications in plants. Specifically, if you talk about stem modification, you will find there are underground stem modifications, there are axillary bud modifications, and also there are certain modifications where they are used for vegetative propagations. So, for this topic, I am going to discuss the modifications that you see on the screen. That is, on the left hand side you are seeing the cactus, while as on the right hand side you are seeing the bougainvillea. Now, yes, so if you see the stem modification, in this modification what happens is, in case of bougainvillea you will find that the axillary positions in the stems have axillary buds, which modifies into a point sharp woody structures that are called as thorns. Here you are able to see these structures present in on the stem. Yes. Okay. So these structures that you are seeing here are called thorns, and these structures are responsible for protecting the plant against grazing herbivores. Similarly, the axillary bud can also modify into slender, thin spiral structures which are called as tendrils. Now, tendrils performs the function of providing support to the stem. You can see these structures in case of gourds family like cucumbers, pumpkins, watermelons. So here the examples will be cucumber, pumpkin, and watermelon. Okay. So these structures can be seen in case of stem modifications. Now similar kind of structures are able to be seen in case of leaf modification as well. So in case of leaf modification you will find tendrils. So this is a cactus. In this you are not able to see any leaves, right? The function of leaves is carried out by the stems itself. That is, they are called as phyloclade. While as the spines that you are seeing here, the pointy structures that you are seeing here are the modifications of leaves. They are called as spines. Okay. So these are also a pointy structures, while as a thorn was also a pointy structure. This, in case of bougainvillea and citrus, you will find these structures are woody, while as they are not so. Okay. While as the function of leaves is carried out by the phyloclade, which is a stem. Okay, the stem modifies itself to perform the process of photosynthesis. Now, why does they need this kind of modification? As you know, in xerophytic conditions or in very dry and hot temperature, the leaves will be a hindrance for the plant because transpiration rate will be very much higher. Okay, so to prevent or to decrease the transpiration rate, what the plants will do is they will modify their leaves into sharp pointy structures so that the surface area of the leaves will decrease and the transpiration of water will also decrease. Okay. So this was the structure. Similarly, the leaves can also the leaves can also modify into tendrils. Now these structures can be seen in P. Okay. Now there are other various modifications that are carried out by leaves, like you will see the Australian acacia, in which the petiole modifies themselves into a thick structure where it performs the process of photosynthesis. Okay, and in some insectivorous plants you will see pitcher plants where the leaves modifies themselves into pitchers. So let's see the question. Thorns found in citrus and bougainvillea are most important vegetative organs for photosynthesis. Are they the most important photosynthetic organs? No, because they are modification of auxiliary bud and they do not perform the photosynthesis. They are rather performs the protection purpose or perform the purpose of protection. Okay. Now the second option is flattened modified stem. So it is not a flattened modified stem. In case of Opuntia, you will see that the stem itself get flattened and perform the process of photosynthesis, which is termed as phylloclade. So this option is also incorrect. Third option is modified axillary bud, which is a correct option. The fourth option says it is a spirally coiled structure. No, thorn is not a spirally coiled structure. Rather, tendril is a modified axillary bud, which is spirally coiled structure. So this option is also incorrect. Is this option clear, or is this question is clear? The correct answer here will be option number 3. Okay. Now let's move to the next question. Now in this question, question number 107, study the pathway given below. So there is a pathway. Tell me which kind of pathway is it? Is it C3 cycle or C4 cycle? Yes? Is it C3 or C4 cycle? First you need to identify the plant, that whether this plant, whether these cells are belonging to C4 plants or C3 plants. So the better way to identify whether the plant is C3 or C4, you can make a vertical section of leaf and find out whether the vascular bundles, if I say this is a vascular bundle, then a vascular bundle should be covered with bundle seed cells forming a wreath. Okay, this is wreath like structure. So this is a characteristic feature of a C4 plant. Now what makes a C4 plant different from C3 plant is that the bundle seed cell have certain features, like this bundle seed cell you are seeing here. This bundle seed cell have certain features, like the cell wall of bundle seed is quite thick and impervious to gases, while on the other hand the mesophyll cell is pervious to gases, and the bundle seed cell have large number of chloroplast. Okay, and the chloroplast here that you are seeing in the bundle seed cell are agranule. That means they does not contain grana. So they are agranule, while as in the mesophyll cell the chloroplast is granule. That means the thylakoid membranes are stacked and they form grana. Okay. Now there are certain features with respect to the presence of enzyme in C4 pathway. You are seeing the mesophyll cell here. Let's see the pathway itself of C4 plant. Okay. So if you see the mesophyll cell, you will find that there is presence of an enzyme called peptase. Okay. This enzyme is only present in the mesophyll cell, while as the bundle seed cell lacks this enzyme. Okay. Now the function of peptase is to fix the primary carbon dioxide, that is coming from the atmosphere. What happens is the peptase will convert the phosphoenol pyruvate, which is the primary acceptor of the atmospheric carbon, so this carbon dioxide will be present from the atmosphere and it will be converted in presence of peptase to form a 4 carbon molecule. Obviously here the 3 carbon molecule phosphonol pyruvate, and one carbon molecule will form a 4-carbon molecule. Okay. Now, what happens? This process is called as fixation, and this A form, the molecule that is formed here is a C4 acid. This acid that is formed by the process of fixation is called oxaloacetic acid. Now, the oxaloacetic acid which is formed in the mesophyll cell is going to be converted into malate or aspartate. Okay. Within the mesophyll cell, this oxaloacetic acid will convert into malate and aspartate. Okay. So, now this malate or aspartate will be transported from the plasmodesmatal connection from mesophyll cell into the bundle seed cell. So, here the acid will be a C4 acid, okay, which can be malate or aspartate, and now the B here is representing that the carbon dioxide is released into the Calvin cycle, right? The carbon dioxide is released from this C4 acid. So, if it was a C4 carbon, okay, and I have removed one single carbon from C4 carbon, how many carbon will be remain here? It will be C3 acid, and the process itself is called as decarboxylation or removal of carbon dioxide. Okay. So, the process here given is decarboxylation. Okay. Now, the process, as you can see here, the presence of pepcase is not present here. Now as the carbon dioxide is released here, the Calvin cycle will occur, and Calvin cycle occur in the presence of enzyme called Rubisco. Now, here there is presence of Rubisco. Okay. Now, Rubisco will accept the carbon dioxide and it will bind with the RUBP and carry out the process of Calvin cycle, where there is utilization of, so in the Calvin cycle there will be utilization of how many ATPs? Yes, yes, chat, how many ATPs? So, in the Calvin cycle, for a single carbon dioxide fixation, we will require 3 ATP and 2 NADPH. Okay. So, this is what the requirement in the Calvin cycle. Okay. So, by the process of carboxylation, reduction and regeneration, now what happens? The amount of ATP here, that is in Calvin cycle, is 3 ATP. But for the fixation or for the enrichment of carbon dioxide into the bundle seed cell require extra ATPs. So, per carbon dioxide we require 2 extra ATPs. So, for this pathway or for this process, C4 pathway requires 2 extra ATP per carbon dioxide enrichment. Okay, in the bundle seed cell. Okay. Plus, obviously here the C3 cycle is carrying on. So, here 3 ATPs is utilized and 2 NADPH is already been utilized. Okay. So, in total 2 and 3, that is 5 ATP is required and 2 NADPH is required. Okay. So, in case of C4 cycle, there is requirement of 5 ATP and 2 NADPH. That's why sometimes C4 cycle is also called as ATP dependent carbon dioxide enrichment cycle, because here the process of enriching the carbon dioxide into the bundle seed cell requires extra ATP. So, that's why most of the higher books say that it is an enrichment cycle. Is that clear? So, after C3 acid that is formed here will be transported again into the mesophyll cell, and the C part here will be, as the C3 acid was itself transported, so C part will be C3 acid, yes, and now this C3 acid will regenerate into phosphonol pyruvate in presence of PEP synthetase. Okay. So, let us see the question. Study the pathway given below and select the correct option for A, B and C. As you already know here that the A is a C4 acid. So the first acid that is formed, or the first product that is formed here, is a C4 acid. That's why the name itself, C4 cycle, is been implemented here. If I am talking about Calvin cycle, the first product will be how many carbon molecule? Yes, if I am telling that here the first product is C4 acid, then the pathway name itself is been C4 acid or C4 pathway. Then in case of Calvin cycle or C3 pathway, the amount of carbon dioxide or the first stable product of the carbon, first stable product will be how many carbon molecule? Yes, it will be C3 carbon. Okay, it will have 3 carbon molecule, that is 3 phosphoglycerate. So here A is C4 acid, which is get transported into bundle seed cell, and process here is carrying out as decarboxylation. And then C3 acid get again transported into mesophyll cell. So C is a C3 acid, and it will regenerate into phosphoenol pyruvate. Okay. So let's see the options. So A is C3 acid, which is incorrect. So first option is incorrect. The second option says that A is C4, okay, and B is a process of fixation. No, it's a process of decarboxylation, where one carbon dioxide molecule has been removed by a C4 acid, which can be malate or aspartate. So this option is incorrect. So third option is C4 acid. A is C4 acid, yes. B is decarboxylation process, yes, and C is a C3 acid, true. So third option is correct, and the fourth option says that it is a C3 acid, which is wrong. So here the correct option will be answer option number 3. Thus the concept of photosynthesis or the C4 pathway is clear to you. Okay. Shall I move? Okay. Let's move to the next question. So the next question is from morphology, where the floral formula is given, and which of the following families has the given floral formula, or which of the following families has the given floral formula. Okay. So these are the families, and we need to identify whether this floral formula belongs to under which family. Okay. So the floral formula is a blessing to you, because the floral formula says a lot about a flower. Okay. So if you are able to understand whether these symbols represents certain, what kind of symbols this all represents for a flower. So let's see one by one. So the circle with plus sign represents that the flower is a radial, a flower is a, just a second, the flower is a actinomorphic flower. Okay. That means it's a radial flower. What is actinomorphic flower? A flower which can be cut into equal part from any other from any vertical plane passing through the center. That is called as actinomorphic flower. Some examples you will see like mustard, datura, chili, that are actinomorphic flower. Okay. Most of the time you will find that some flowers floral formula have representation of percentage sign. This represents the symmetry of flower of zygomorphic. Flower. This is a bilateral symmetry, where a flower can only be divided into 2 equal halves when we cut the flower into single vertical plane from the center. Okay. So the example here will be Fabaceae family, where pea, bean, and you will see gulmohar, right? So these are the zygomorphic flowers. Now this symbol represents the sexuality of the flower. That means a flower can be bisexual, that means it contains both androecium and gynoecium in the single flower, or can contain either androecium or gynoecium. So for the male sign we represent this particular symbol, and for the female we represent this. Okay. So it is if I am representing a flower in which a floral formula represents this kind of symbol, then it is a male flower, and this is a female flower, and it is a bisexual flower. Okay. Now the K here represents the calyx. Okay. So the K here represents the calyx. Now, calyx is the outermost wall of a flower. It is responsible for protecting the bud of a flower. Okay. So calyx have a unit, a single unit of a calyx is called sepal, and the sepals can be free or can be united. Okay. So if a sepal is free, then it is called as polysepalous condition. And if it is fused, then it is called as gamosepalous condition. Okay. So for polysepalous condition, the representation can be n, or number of flowers, units, or for gamosepalous condition you can use brackets. Okay. For numerical digit, you will cover the numerical digit into brackets. Okay. So here you will see that the flower have which condition, poly or gamo? Yes, the condition here is gamosepalous. Okay. Now the C represents the corolla. Okay. Now corolla are colorful. Okay. Now corolla are generally colorful, and they are responsible for attracting the insect toward the flower so that the flower can get pollinated. Okay. Now one question arises, you have seen bougainvillea, right? Right? You have seen bougainvillea. In bougainvillea you have seen in the previous slide itself, you have seen this bougainvillea, right? Tell me, is it a sepal or a petal? Yes? Is it a sepal or a petal? So many a times what happens, a flower which is quite small is not able to be visualized by the insects. So flower modifies themselves, flower modifies their sepals into colorful and very vibrant colors so that an insect can get attracted toward itself. Okay. So this is a sepal. In case of bougainvillea, it's a sepal. So let's see the concept. Okay. So now, so corolla is made up of units that are called as petals, and now in as the same case in sepals, petals can be free or fused. Okay. So petals can be gamopetalous, that is fused, or can be free, that is polypetalous. Okay. So if I am saying a corolla is here, the condition is gamopetalous. Okay. So the representation will be like this, and it is, if it is poly, then the representation will be like this. Okay. Now the A represents in a floral formula represents the androecium, and androecium consists of stamens, and stamens is made up of filaments and anther. Okay. So A represents the androecium. Okay. And they have a unit that is called as stamens, and stamens consist of filaments and anthers. Now stamens can also be grouped in a bundle, so they are called as monoadelphous, or can be in two bundles, that is diadelphous, or can be in multiple bundles, or can be in more than two bundles, so they are called as polyadelphous conditions. Okay. So most of the times you will find that a stamen is bind with the other wall, that is corolla. In case of this floral formula, you will find that this symbol represents that the anther is attached to the corolla. That means it is a epipetalous condition. Okay. But sometimes you will see that a flower doesn't contain any calyx or corolla, it contains perianth, which is represented by P. And in this case, if an anther is attached to the perianth, then the condition is called as epiphyllous. Okay. And this condition is generally seen in some flowers where the perianth condition is present. Okay. Now here the condition is normal without any bundle, so this is anther free. Okay. Now you will see the gynoecium here, which is represented in the form of G. Okay. And you will see the line underline here. This underline represents that the other floral parts are present below the gynoecium. That means the flower itself is a hypogynous flower, or the gynoecium is superior. Okay. And the condition here is fused. That means it is syncarpous condition. So it's represent gynoecium, and it can be fused. If it is fused, the carpels, if they are fused, then the condition is called as syncarpous, and if it is free, then the condition is called as apocarpous. Okay. So this much information is being stored in this small floral formula. So this floral formula represents the family of Solanaceae. Okay. So let's see the question. Which are the given, this floral formula belongs to Solanaceae family. So first option is true. While as in Fabaceae, you will see zygomorphic flower, not actinomorphic flower. So we can cancel out by this knowledge that it have zygomorphic flower. In the third option, Liliaceae have perianth condition, not calyx or corolla condition. So it have this condition, so it can also be ruled out. And Brassicaceae can be remembered by the condition where the sepals are in two walls, or in this kind of arrangement. Okay. So this is what you can use the knowledge of all the floral formulas and identify whether this particular floral formula represents which family. Okay. So here the answer is 1. Let's move to the next question. Okay. So the next question is the cell envelope. So this question belongs to cell, the unit of life, and the cell envelope is generally seen in the bacteria, okay, in the groups of Monera, where particularly bacteria. So you will find in bacteria there is various complex cell envelopes. And cell envelopes are bound by 3 layers. Okay, which comprise of glycocalyx, which is the outermost part of the outermost part of the cell envelope. Then comes the middle layer, which is the cell wall, and the innermost layer, which is plasma membrane. Though these all structures have different functions and different structural integrity, but they together act as a unit for protection, or together act as a unit, as a protective unit for the bacteria. Okay. Now the cell envelope itself, if you are seeing here, the capsule that is represented here, the cell wall and the plasma membrane, they together are termed as cell envelope. Okay. If you are able to see the yellow color, then this is what represents the cell envelope. Okay. Now on the basis of the cell envelope, the bacteria can be classified by gram staining. If a bacteria takes up the gram stain, then it is gram positive, and if a bacteria doesn't take the gram stain, then it is gram negative bacteria. Okay. So here the cell envelope provides the identity to the bacteria, that whether on the basis of the gram staining, whether the bacteria is gram positive or gram negative, and also it provides the protection against immune system if you talk about the overall structure. Like in case of glycocalyx, you will see glycocalyx is a quite complex material which can be thick, okay, or can be slimy layer. So if it is a slimy layer, then if it is loose sheet, then it forms a slimy layer. Okay, and if it forms compact and thick and tough structure, then it forms capsule. Okay, now capsule has a certain property. Okay, you have seen the experiment of Griffith. Yes, everyone have seen the experiment of Griffith where he used the S strain of pneumonia and R strain of pneumonia. Okay, the rough strain or the R strain was not able to infect the mice, while as the smooth strain was able to infect the mice and cause pneumonia, and ultimately the mice died. So the smooth strain have the capsules. So it means the capsule helps the bacteria to escape the host immune system. So that's one of the property of glycocalyx. Okay. Yes, Kumari, what do you have? One question, please ask. Okay. So this is what glycocalyx is responsible for. Now the cell wall provides the structure to the bacteria, and it also provides protection. Okay. And the plasma membrane is quite similar to the eukaryotic cell, and it is semi-permeable. So it is the membrane which is in the contact of the outer world. Okay. So let's see the options. Yes, Kumari, I am still waiting. What's your question? So the cell envelope is present only in eukaryotic cell. No. The cell envelope consists of tightly bound 3-layer structure with plasma membrane being the outermost. So this option is incorrect because plasma membrane is the innermost layer of cell envelope, not the outermost layer. Okay. Third option is has 4 layers performing similar function. Okay. Kumari is asking whether is capsule hard slimy layer, I mean how it forms. Okay. So not hard slimy layer, it can be loose sheet of glycocalyx. Okay. So it can be a loose sheet of glycocalyx that forms a slimy layer, or it can be quite hard and tough to form a capsule. Okay. Okay, Kumari. So third option is has 4 layers performing similar function. Has 4 layers. I have told recently that it have only 3 layers. Okay. So this option is incorrect. And fourth option is acts as a protective unit. Yes, the cell envelope, it as a complete unit acts as a protection unit. Okay. So here the fourth option is the correct one. Let's move to the next question. Question number 102. Read the following statements and choose the correct option. So here comes the assertion and reason question, where most of the time students are very much confused about whether a reason is a correct explanation of the assertion or not. Okay. So there is simple trick to see whether the reason is correct for the assertion or not. You can use because, hence, or therefore. Okay. These statements can help you to analyze the statements and then re-evaluate whether these reasons are correct explanation of assertion. So let's see the assertion. Mitochondria can synthesize some of its own proteins. Okay. So this statement is true, right? Because mitochondria consist of ribosomes. Now tell me what kind of ribosomes are present. Is it 70S or 80S type of ribosomes that are present in the matrix of the mitochondria? Yes. So you will find that the mitochondria matrix contains 70S type of ribosomes. Okay. Now you know ribosomes are responsible for the process of translation, and from the translation you will synthesize the polypeptide chain which can convert into enzymes. Yes. So they can synthesize their own proteins. Just a second. So they can synthesize their own protein. So they are responsible for translation, and after translation they will form polypeptides, or polypeptide can form enzymes. So indirectly they are able to translate their own enzyme. But not all enzymes are being synthesized by the mitochondria. They do need the ribosomes that are present in the cytoplasm of the cell. Okay. Now, the matrix also contains circular double-stranded DNA. And this feature, that is circular double-stranded DNA and presence of 70S ribosome, is quite similar, right? If you consider the prokaryotic cell, because prokaryotic cell have also the same features, that is 70S ribosome and circular double-stranded DNA. Is it correct or not? Right? It's correct. So that's one feature that is very much peculiar about mitochondria. Similarly, the other organelle that you see in plants, that is chloroplast, have the same property, that is they also have 70S ribosome and the circular double-stranded DNA. Okay. So first statement, that is assertion, was true here. Now the second statement says that both inner and outer membrane of the mitochondria have enzymes. So if you see the organelle, you will find the outer membrane and inner membrane. Okay. And this both membranes contain their own enzymes and they have their own different functions. Okay, if you have seen the process of respiration, you have learned the oxidative phosphorylation process. And in the oxidative phosphorylation process, you have seen the enzymes like NADP reductase, succinate dehydrogenase enzymes, right, which are present in the inner mitochondrial membrane. So that means the inner mitochondrial membrane contains the enzymes, whereas the outer membrane also contains certain different enzymes. Okay. So the statement, that is reason, here is true. But is it explaining the reason or is it explaining the assertion? Let's see this. So both inner and outer membrane of the mitochondria have enzyme. That's why mitochondria can synthesize some of its own protein. So thus the enzymes present on the outer membrane is also responsible for the production of proteins in the matrix. No. So here the assertion is not quite explaining the reason. If a reason was something like this, that the mitochondria can synthesize some of its own protein because mitochondria have its own ribosome or own 70S type of ribosome, then it may have been a correct or somewhat logical explanation of the assertion. Okay, but the reason is not explaining the assertion here. So here reason is assertion and reason are true, but reason is not the correct explanation. So here first option is incorrect, while is the second option, both A and R are true, but reason is not the correct explanation. So this is the correct option, while as 3 and 4 are ruled out. Okay. Okay. Now let's move to the next question. Now comes the most important question that has been asked in NEET a lot of times on the basis of scientists or their discoveries in the process of photosynthesis. And most of the time students are quite confused whether a scientist that are seen on the screen have made this discovery or not. So to memorize this concept, let's take the history and re-evaluate what are the contribution of each scientist one by one. Okay. Are you ready for the history? Let's see. So around 1770, Joseph Priestley, who used the bell jar and a candle and a mice. Okay. So Joseph Priestley used a bell jar, candle and a mice to identify whether why the candle, or to identify his experiment. He was intrigued that when the bell jar contains the candle got extinguished after a certain while. When the mice was kept in the bell jar, the mice got suffocated and died. And then he implemented a plant. Okay, that plant was a mint plant. Okay, so when he input the mint plant and the candle in the same jar, he find out that the candle was not extinguished after a certain while. Similarly, he got the same result for the mice, where the presence of mint plant and the mice, so mice was able to survive. So he made a hypothesis that there is something about the plant that is replenishing the air, whatever the breathing animals or the burning candle removes. Okay, that was his hypothesis, which was quite true. Okay, but he didn't know whether this air or the compound that is released by the animal itself or the candle itself was what, carbon dioxide, oxygen, or anything. Okay, and what was the plant releasing. Okay, but though Joseph Priestley in 1774 identified the or discovered oxygen. Okay, so this was what the hypothesis that was given by Joseph Priestley. Now after Joseph Priestley came Jan Ingenhousz. Jan Ingenhousz took the same experimental setup of Joseph Priestley, but he made one major change, and that was keeping the experimental setup in the light condition and in the dark condition. Okay, so what happened when he find out that in the light condition, when the experiment was kept in the light condition, when the experimental setup of the Joseph Priestley was kept, then the result was same as the Joseph Priestley have predicted. But in the dark condition, the experimental setup was failed to express the same result as in the case of Joseph Priestley. So what was the reason behind it? So to make a possible or logical explanation, he used a aquatic plant, which is hydrilla. He used the aquatic plant and kept the aquatic plant in the light condition and in the dark condition. He was very much intrigued why in the light condition the hydrilla green parts were removing bubbles in the water, while as in the dark condition, so here bubble was able to form, while as in the dark condition no such bubbles were being produced by the plant. After certain while, he find out that the bubble was the oxygen. So he state that light is important for the process of removing certain for removal of oxygen. Okay. Yes, Kumari, hydrilla is most of the times been asked in the NCRT, in the NEET. So it's quite important. Okay. So he said, or he hypothesized that light is important for removal of oxygen. After Jan Ingenhousz came Julius von Sachs, and in the year of approx 1854, he said that the green part of the plant, here, the green part of plant was able to synthesize glucose. Okay. So the green part was responsible for production of glucose. That was his experimental proof. Okay. Now after Julius von Sachs came another scientist in around 1882. He made a breakthrough result where he find out that when he used cladophora, a unicellular filamentous green algae, okay, when he used the cladophora and aerobic bacteria along with the light spectrum that was split by the help of along with the light that was split by the help of prism, he find out that in his Petri disk the hydrilla the cladophora that was present here, okay, and in the suspension aerobic bacteria were grown, okay, and he found out that in the light spectrum only the red and the blue light region is the location were the location where large amount of aerobic bacteria were growing. That means that here the process of removal of oxygen was quite much higher as compared to the other light spectrum. And through his experiment, the first action spectrum of photosynthesis was described. Okay. After T.W. Engelmann came Cornelius van Niel. So up till now, up till this experiment, we are quite much clear about the reaction of what's the reaction in the photosynthesis, right? So CO2 is required, water is required as in the plant, and the carbohydrate will be formed, and there will be liberation of oxygen. Up till now, from this and this year, up till now, we have a somewhat formula for the process of photosynthesis. But scientists were still confused whether the oxygen that is coming from the process of photosynthesis, is it coming from the molecule of carbon dioxide or is it the H2O itself? So to clarify this concept, Cornelius van Niel, who was a microbiologist, used purple and green sulfur bacteria which uses H2S instead of H2O as a hydrogen donor. So what happened after the process that is carried out by the purple and green sulfur bacteria, he find out that the sulfur was released in the atmosphere. Similarly, like the oxygen was released during the process of photosynthesis, sulfur was releasing in the atmosphere. So he concluded that the oxygen that was being removed by the process of photosynthesis is by the H2O, not from the carbon dioxide. That means he made a hypothesis where H2O is splitting and converting into oxygen, which you will see in the chemiosmotic hypothesis or oxygen evolving complex. Okay, so then after that, all this Mahabharata and Ramayana, the concluding reaction of photosynthesis was requirement of carbon dioxide, requirement of water, and production of carbohydrates and oxygen. Okay, this was the last conclusion of the process of photosynthesis. So now let's see the question. Choose the correct pair with respect to important discoveries in photosynthesis. Okay, Julius von Sachs, sunlight is essential for photosynthesis. Julius von Sachs were responsible for identifying the green part of the plant was responsible for glucose synthesis. So this option is incorrect. Second option says Jan Ingenhousz, H2S is hydrogen donor in purple and green bacteria. No, it was in the year of 1931, that is Cornelius van Niel, who discovered that H2O is the hydrogen donor, H2S is the hydrogen donor. So this is also incorrect. Third option says Engelmann, first action spectrum of photosynthesis was described. So yes, by the experiment of Engelmann, the first action spectrum of photosynthesis was described. And the fourth option says van Niel, glucose is produced during photosynthesis. So it is incorrect, as it was Julius von Sachs. Okay. So this concept is clear to you? Yes? Okay. Now the last question here is question number 110. Select the incorrect statement with respect to tapetum. Now tapetum is the anther wall layer. Now to understand this concept, you need to understand first where is the tapetum located and what are the other layers that are present in a anther wall. So let's see. So this is a section of anther wall layer. So the outermost layer here you are seeing, which is single layer, called as epidermis, which is responsible for protection of anther wall, for the protection of anther. While as you will see some 1 to 3 layer, we will see some layers that are called as below the epidermis, called as endothecium. Okay. Now endothecium are made up of alpha cellulosic fibrous bands, which are able to absorb water and responsible for a dehiscence of anther. Below the endothecium, you will see some 1 or 3 layers of middle layers. These middle layers are ephemeral. That means they are going to get degenerate after the maturity of anther. Okay, and the innermost layer you will see tapetum, and the tapetum have certain features, like the cells of the tapetum are dense and they have dense cytoplasm, and they have multiple nucleus. Okay, so they are polynucleated. And this layer or the tapetum layer surrounds the sporogenous tissue and also is responsible for the nourishment of the sporogenous tissues. Okay. The outer three wall layers, that is epidermis, endothecium and middle layer, are responsible for protection and dehiscence of anther. Okay. So this is what the detailed structure or each layer structures is seen here, right? In the middle of the microsporangium, you will see a compact cell structures which are called as sporogenous tissues. And these compact cells are able to produce pollen grains during the maturity of the anther. Okay, so let's see the options. Select the incorrect statement. Select the incorrect statement with respect to tapetum. First option says that tapetum is the innermost wall layer of anther. Okay, this option is correct. Second option says it surrounds the sporogenous cells of anther. This is also true. Third option says it performs the function of protection and helps in dehiscence of anther. No, the outer three wall layers of the anther performs this function. So this option is incorrect. And the fourth option says it nourishes the developing pollen grains, which is true. So fourth option is true. So here we have asked about the incorrect statement. So here the correct answer will be option number 3. So I hope this session was quite fruitful to you, and all the best for the upcoming NEET exams, and stay focused, stay hydrated. Thank you.